Hi everyone. In today's video, we're going to be talking about experimental design. So how we set up an experiment and what the various variables and controls mean in an experiment. So let's get started. Okay, so let's say hypothetically we wanted to see what would the best liquid to give a plant to see how it would grow. What would we need to do to design this experiment? Well, the first thing that we would need to think about is if we're trying to see what is the best liquid to give a plant, well, we have to have a baseline of what a plant needs to grow. So for our baseline, we would probably give the plant water because we know it is a liquid that plants need to grow. And then we would probably test other liquids to compare to how well the plant's doing. So we could test things like juice, things like soft drink, and etc., etc. So looking at this right here, we've already kind of defined two key groups when we do an experiment. Our control and our testing. So our control is basically the experiment that has all of our variables to the best of our ability kept constant so that we can see a particular reaction. Our testing group or our testing variables are things that we alter based on our control as our flat line and to use a comparison. So whenever we do an experiment, we need to have one group of the experiment that is the bare minimum that whatever we're testing will work. And then we need to look at what if we just change one variable and compare it to our baseline? Because we always need to have something to compare our experiment to see did it work or did it not work. So all science experiments have that basic dichotomy in the sense that we have one experiment, which is our, if we are doing nothing, this is what should happen. And then if we've changed one thing, these are what we compared to our original to see if there is a significant difference or not. Which gives us to our first term of our testing scenario, is basically what is your experiment trying to figure out. And if we're looking at our last example as our example in this case, we're trying to figure out what liquid causes our plant to grow the best. So our testing scenario is really what are you trying to do? So if we're looking at this right here, A and B, this would be our testing scenario. And C would be our control. Because we need to always compare something to something else. So testing scenario, which liquid would be the best to have our plant grow? And our control is what we are comparing our testing scenarios to. So when we're talking about a control, it's things that we are not changing in the experiment, things that we are trying to keep constant. Now, there is only so much you can do to try and keep constant. So even when we're looking at our control variables, sometimes there are things that can change. So if we were doing an experiment outside and there's certain things that you cannot control. You cannot control the weather, you cannot control the temperature. So those are things that are out of your control. But if we were doing an experiment indoors, well, then there are things that we can control. We can control the temperature in the room. We can control the humidity in the room. We can control the amount of light in the room, and so on and so forth. So anytime you have a control variable is things that you are not changing. Now, we can break up our controls into two categories, positive and negative control. And the term positive and negative control has to do with the outcome of that trial. So positive controls are variables that you know are going to cause the experiment to work. So if we're talking about the plant, right? So giving the plant water would be a positive control because I know plants need water to grow, so it's my base liquid that I can be giving my plant, and I know that my plant will grow giving it water. A negative control is the opposite. It's variables that you know that are going to cause the experiment to fail. So if we're talking about the plant, so if I have a trial where the plant is given no liquids, well, that would be a negative control. I should expect the plant to die. So depending on the experiment, you might have positive controls where you know this is the base minimum and I know that my experiment is going to work with the base minimum, and you're using that to compare to see if whatever you are changing will cause the experiment to perform better or to perform worse. On the flip side, a negative control is, okay, I know this will cause my experiment to fail. Now, if I change that one variable, is my experiment going to be successful and also, or is it going to also fail? So depending on how you set up your experiment, you could have just a positive control, you could have a negative control, or you could have a scenario where you have both a positive and a negative control. It really does depend on what your experiment is. Usually when we're doing experiments with living organisms, we tend to not want to have negative controls because it tends to usually kill the living organism, and we don't like doing that. So if we're talking about our plant, some control variables would be the same plant species, right? So if we're doing an experiment where we're trying to see what liquid is going to cause the plant to grow the best, well, we would want to do it on the same plant species. And then once we've figured it out for one species, test it on another species and see if it's the same liquid for different species. So that would be one. The same soil for the same argument. Not all plants could live in the same soil types, so you want to make sure that the soil would be consistent for all of them. Also, the same amount of sunlight. We know that plants, because they photosynthesize, need sunlight. So because we are not testing whether the plant can produce glucose or not, we're just basically testing what liquids are the best at helping them produce glucose. We want to make sure that the sunlight is the same. So these would be some of the variables that you would want to control for. Now, your independent variable. This is probably the key variable when we're talking about an experiment, because your independent variable is the one variable that you are changing compared to your control experiment. So when you're actually designing your experiment, your independent variable is the one thing that you are changing, because when we're doing an experiment, you cannot change more than one variable at a time, because if you do, whatever result you get, you have no idea if it's due to the one variable, due to two variables, or due to a combination of them. So you can only change one at a time. And your independent variable is that one variable that you change. The dependent variable is what you are measuring to see if a change occurs. So your dependent, as the term sounds, always relies on the independent. So if we're talking about our plant, right, if we're going back to our plant, our independent variable is the liquid, right? The type of liquid that I'm giving the plant is the independent variable. What I am measuring to see if it has an effect on the plant is my dependent variable. And in this case, I am measuring plant height, right? Because we're saying that if the liquid has a positive effect on the plant's growth, the plant will grow and grow tall. If the variable has a negative effect on the plant's health, well, the plant won't grow, or the plant will grow very, very short compared to the control. So your dependent variable is always the thing that you are measuring. So whether you are measuring reaction time, whether you're measuring height, whether you're measuring mass, it doesn't matter. It is what are you measuring to see if there is a noticeable change in your reaction. So let's talk about this hypothetical experiment. So let's say that you are trying to make an experiment to see if drinking Gatorade will improve your golf game. Well, this right here is your testing scenario. Right? And your testing scenario, when we're talking about the scientific method, is your problem. What are you trying to find? And in this case, we are seeing, does drinking Gatorade improve my golf game? Now, for those who don't know, in golf, the lower your score, the better your game is. So if we're trying to do an experiment to see if Gatorade is improving your golf game, well, we're going to be measuring golf scores. And golf scores that are low compared to our control would say that yes, Gatorade helps. Golf scores that are higher compared to our control would basically say no, Gatorade does not help. And if after the experiment we find out that the golf scores with drinking water and the golf scores without drinking water are the same, well, then there is no noticeable difference between Gatorade and another liquid. So if we were golfing for 10 times over a span of 2 weeks and we played one game of golf at the same golf course every single time of the day, all of these would be our controls. They're things that we are keeping the same between both of our experiments. The first week, we only drink water. So only drinking water, this would be, again, the control scenario. The second week, only drinking Gatorade, well then Gatorade right here is our independent variable. It's the one variable that differed from the control scenario. So we noted that when you only drank water, the golf score was 90. When you only drank Gatorade, the golf score was 78. So if we're looking at seeing if Gatorade had an effect or not, well, we need to look at the results from the control. So control, again, was just with water. And we see that the average score is 90. So if we're looking at the experiment, so with Gatorade, we see that the average score is 78. And we can see that there is a substantial difference between just water and Gatorade. So if we were looking at what would be our conclusion from this experiment, we would say that Gatorade does improve the score. Now, obviously, this is just a hypothetical scenario. There's a lot of other variables that you have to look at. But if we're looking at this from a basic setup, it would be this. So what I'd like you to do now is I'd like you to pause the video, read the scenario, and try to see if you can figure out what the problem is, what the control is, what the independent and the dependent variable are. Once you've done that, unpause the video and we'll go through the answers. So if we're looking at this right here, our problem is basically to see if greenhouses improves the plant's growth. Now, if we're looking at our control, well, we have basically the same type of rows. And our control situation would be the rows growing outside. Our independent variable would be the rows growing in the greenhouse. And our dependent variable, well, what we're looking to see if there is an effect is the number of flowers on each rows. So again, our control scenario and our independent scenario is what is the one thing that differs between both setups. Now, because we're testing to see if roses grow better in a greenhouse, well, our independent variable is the rows growing in the greenhouse. Now, our control variable is the rows growing outside because we're trying to see if growing in a greenhouse is better than growing outside. So that's why that's our controlled variable. And again, our dependent variable is what we can measure to see if this makes an improvement, yes or no, which is the number of flowers on each rows. Okay, so again, what I'd like you to do is to pause the video, read this scenario, and then try to figure out what the problem, what the control, what the independent and dependent variable in this scenario is. But I'm just going to read it for you before you pause. So you have 4 sunflower plants. 3 are given water with different salt concentrations in them. And the last one is given fresh water. They're watered daily at the same time and are in the same room. After 2 weeks, the height of each sunflower is measured. So pause the video and again, try to figure out what the problem is, what the control scenario is, what the independent and dependent variable is. Okay, so in this experiment, the problem is, does salt water improve plant growth? Right, because that's what we're trying to see. Does salt water in varying concentrations of salt water improve my plant growth? So my control scenario is going to be the sunflower given water. Right, because we know plants grow with water. So our control variable is, okay, so I'm just giving my plant water and we're going to see that sunflower is going to grow a certain amount. And then we're going to use that as our baseline comparison to all of the other sunflowers that are given salt water. Now this control is a positive control because I know plants need water to grow. So by giving my sunflower just water, it will grow. My independent variable would be my various saltwater concentrations. Because again, that is what I am differing from my control, which is just the plant getting water. And then my dependent variable, again, would be the height of the plant. So I hope this helps going over some of the key terminology in an experiment. And these are things that whenever you're looking at any scientific experiment, well, that's basically what they're trying to do. They're trying to see, does changing this one variable have a positive or negative effect? And to be able to compare if there is a positive or negative effect, well, we always need to run one trial of the experiment that we know will either work or we know will either not work. And by changing that one variable, we can compare it to that. Have a great day.